So Direct Corporation is a biopharmaceutical company that's leveraging novel epigenetic regulator therapeutics in order to treat chronic and acute organ injury. And we also manufacture and globally sell ALZET Osmotic Pumps, which are a preclinical tool used to induce disease states in preclinical models and to deliver therapeutics. And Direct has had ALZET pumps as part of its portfolio for many years. So ALZET pumps are an implantable drug delivery device that researchers use to deliver therapeutics. They would use a syringe to fill the pumps, implant the pump in the back of the animal, and it doses automatically and continuously for as long as 6 weeks, single pump. ALZET pumps come in 12 different models, and within that are 3 specific physical sizes. The smallest is a 100 microliter pump that can be used in an animal that's at minimum 10 grams in body weight. And then we have a 200 microliter size and a 2 milliliter size. All of those can be used in rats and larger models as well. Researchers really need reliable tools that consistently perform so they can focus on the scientific question. They need tools that don't add confounds to the study so that might cloud the research results. And ALZET pumps have a proven track record with over 21,000 published studies in the public domain. Researchers also really appreciate the automated dosing, so that means they don't have to come in nights and weekends to inject animals. And that also means that the animals are not subjected to the stress and to the behavioral confounds that come from that kind of handling. ALZET pumps are used in neuroscience research, in cancer research, cardiovascular, endocrinology, orthopedic, pharmacology research. Really, since the pumps are sold empty, the sky's the limit when it comes to the different areas that the pumps can be used in. ALZET osmotic pumps contribute to cardiovascular research in 2 main ways. One is delivering an agent that sets up a model of disease, and the other would be delivering an agent intended to reverse a disease process, either a therapeutic or an agent that confirms the role in that disease of a particular target, which could be a proxy for a future therapeutic. So in modeling disease states, some examples would include hypertension, where the ALZET pump is used to deliver something like angiotensin II or aldosterone to establish a model of hypertension. Another example would be abdominal aortic aneurysm, also using ALZET pumps to deliver angiotensin II. And a third would be a model of heart failure, such as using isoproterenol delivered by ALZET pumps. And then considering using pumps to reverse some pathological process, some recent examples might be in coronary artery disease, delivering 5-HT2 receptor agonists. Another might be in a model of cardiac hypertrophy, delivering agents that affect vascular smooth muscle cell phenotypes, such as, for example, FGF23. Lastly would be in cardiac ischemia, in which ALZET pumps might be used to replace endogenous peptides in various knockout models to identify cardioprotective mechanisms in post-myocardial infarction remodeling. So one common misconception about the ALZET pumps is that the molecular weight of the drug substance that's being delivered influences the release rate from the pump. But actually the release rate from a given pump is set at manufacture. And so the researcher is filling the pump with a drug solution, and the pump operates at that predetermined release rate. So the ALZET pumps, once implanted, are well tolerated. Many people are studying behavior or motor activity and are able to perform those tests without a significant confound from the implant itself. So the benefits of using ALZET pumps are especially that you can implant the pump and leave it to dose in an automatic fashion without having to handle the animals frequently during the study, such as you might need to do with frequent injections. Also compared with other methods of delivering drugs, with many common methods, you get really fluctuating plasma levels. And with the ALZET pumps, the idea is that it maintains a constant level in the animal so that you're able to see the effects of the compound developing. And also, many methods for dosing animals induce a lot of stress, not just injection, but also gavage and other methods cause more stress to the animals than by the implantation of the pump and then being able to be hands-off for the rest of the dosing period.